There is an animal in the ocean tonight that has 3 hearts, blue blood, and a brain that extends into each of its 8 arms. An animal that can taste everything it touches, see color despite being colorblind, and solve problems that stump creatures with far larger nervous systems. The octopus has been quietly extraordinary for 500 million years, and almost everything about it defies the categories we use to understand animal life. It is soft-bodied, but not defenseless. It is short-lived, but not simple. It is solitary, but not unaware of others. Tonight we will follow it gently, from the strange architecture of its distributed mind, through the skim that changes faster than thought, into the deep questions its existence raises about intelligence itself. So, let the room soften around you now. Let your breathing slow to the pace of something patient and unhurried, and drift down with me into the cold, dim water, where the most alien mind on our planet is already awake, already watching, already thinking in ways we are only beginning to understand. The octopus nervous system is organized in a way that has no real equivalent among vertebrates, and understanding it requires setting aside most of what we assume about how a brain and body are supposed to relate to each other. A human brain controls the body through a clear hierarchy. Signals travel down the spinal cord and out through peripheral nerves to muscles, with the central brain making most decisions and the peripheral nervous system largely executing them. The octopus does something fundamentally different. Of the roughly 500 million neurons in a typical octopus, only about 50 million reside in the central brain. The remaining 450 million are distributed across the 8 arms themselves, with each arm containing its own local neural network, capable of processing sensory information and generating movement patterns independently of the central brain. This means that when an octopus reaches an arm into a crevice to search for food, the arm is not simply executing instructions relayed from above. It is making decisions on its own. The local neural network in the arm can direct the suckers, adjust grip strength, interpret tactile and chemical signals from the surface being explored, and modify the search pattern in real time, all without waiting for input from the central brain. The central brain, meanwhile, can be attending to something entirely different, perhaps monitoring visual input from the eyes or coordinating the behavior of another arm. The octopus is, in the most literal neurological sense, doing several different things simultaneously with different parts of its nervous system, in a way that feels closer to a colony of semi-autonomous agents than to the centralized command structure of a vertebrate. The independence of the arms has been demonstrated strikingly in experimental settings. Researchers have shown that a severed octopus arm, no longer connected to the central brain, will continue to respond to tactile stimulation, perform gripping movements, and even attempt to pass food toward where the mouth would be for up to an hour after separation. This post-separation behavior is not the simple reflex activity of a few remaining firing neurons. It is coordinated, goal-directed movement generated by the arm's own neural network operating autonomously. What this tells us about the octopus's everyday experience of having 8 such arms, each with its own processing capacity running in parallel, is a question that neuroscientists find genuinely difficult to frame, let alone answer. The central brain of the octopus is itself highly organized, despite its relatively modest size. It consists of approximately 50 lobes, each associated with different functions, ranging from visual processing and memory to the coordination of arm movements and the integration of information from the chemoreceptive suckers. The optic lobes, which process input from the two large eyes, are particularly prominent, reflecting the octopus's heavy reliance on vision for hunting, navigation, and communication. Immediately adjacent to the esophagus, which passes through the center of the brain in a ring-like arrangement, are the vertical and superior frontal lobes, structures associated with learning and memory consolidation in a region of the octopus brain that shows some functional parallels to structures involved in memory formation in vertebrate brains, despite having evolved those capabilities entirely independently. One of the more philosophically interesting aspects of octopus neuroscience is what the distributed architecture implies about the animal's subjective experience, if it has one. If most of the decision-making is happening locally in the arms rather than centrally in the brain, is there a unified self experiencing and coordinating all of this? Or is the octopus something more like a loosely federated community of semi-independent processors with a central coordinator? Researchers studying octopus behavior have noted that the animal does appear to act with unified purpose when needed, coordinating multiple arms toward a shared goal in ways that suggest central integration. But in exploratory or low-priority situations, the arms seem to operate with a degree of independence that blurs the boundary between one animal with 8 arms and 8 agents with one shared body. The evolutionary path that produced this extraordinary neural architecture is itself interesting to consider. Octopuses share a common invertebrate ancestor with animals like clams and snails, whose nervous systems are generally modest in size and capability. The cephalopod lineage, which includes octopuses, squids, and cuttlefish, underwent an apparently rapid expansion of neural complexity, developing large brains, sophisticated sensory systems, and complex behavior patterns independent of any input from the vertebrate lineage. The octopus and the vertebrates represent 2 entirely separate evolutionary experiments in building a complex, behaviorally flexible nervous system from invertebrate foundations. And the fact that both experiments produced animals capable of learning, memory, problem-solving, and what appears to be genuine curiosity is one of the more thought-provoking facts in all of comparative neuroscience. The octopus circulatory system is built on a different chemical foundation from our own. And the reason for that difference leads directly into the chemistry of oxygen transport in cold water, which turns out to have consequences for nearly everything else about how the octopus body works. Human blood is red because it uses hemoglobin, a protein built around iron atoms, to carry oxygen through the bloodstream. When hemoglobin binds oxygen, the iron-oxygen complex reflects red light. Octopus blood is blue because it uses a different oxygen-carrying molecule called hemocyanin, built around copper atoms rather than iron. When hemocyanin binds oxygen, the copper-oxygen complex reflects blue light. The color is not incidental. It reflects a fundamentally different solution to the same respiratory challenge. Hemocyanin is dissolved directly in the blood plasma rather than packaged inside red blood cells, which gives octopus blood a distinctive, slightly viscous quality. In terms of oxygen-carrying efficiency, hemocyanin is generally less effective than hemoglobin at warm temperatures, but it performs better in cold water and low oxygen environments, making it better suited to the deep, cold, often oxygen-limited habitats that many cephalopods occupy. Some researchers have also noted that hemocyanin releases its bound oxygen more readily when blood acidity changes, a property that may help octopuses cope with the rapid changes in metabolic demand that occur during active hunting or escape from predators. The blue blood is not a biological compromise. It is an adaptation precisely calibrated to the octopus's chemical and thermal environment. The three-heart arrangement of the octopus circulatory system serves a specific anatomical purpose. Two branchial hearts, positioned at the base of each gill, pump blood through the gill tissue where it picks up oxygen and releases carbon dioxide. The oxygenated blood from both gills then flows to the single systemic heart, which pumps it through the rest of the body. This division of labor between the branchial hearts and the systemic heart allows the oxygenation step and the distribution step to be handled separately, with each optimized for its specific task. The branchial hearts can generate the pressure needed to push blood efficiently through the dense capillary network of the gills, while the systemic heart generates the sustained pressure needed to drive circulation through the whole body, including the arms. One of the notable consequences of this three-heart system is what happens when an octopus swims. Octopuses typically move by crawling on their arms, which is energetically efficient. When they need to move quickly, they switch to jet propulsion, drawing water into the mantle cavity and expelling it forcefully through a siphon to generate thrust. This jet propulsion is effective for short bursts of rapid movement, but is metabolically expensive, and interestingly, compromises the function of the systemic heart. When the octopus contracts its mantle for jet propulsion, the mechanical compression of the body cavity partially impairs the systemic heart's pumping action, reducing blood pressure in the body's circulation at the very moment when the muscles need maximum oxygen delivery. This is why octopuses become fatigued relatively quickly during sustained jetting and prefer walking to swimming for routine movement. The metabolic rate of octopuses is notably higher than that of most other mollusks, reflecting the energetic demands of a large, active nervous system and a body that is in continuous muscular adjustment. An octopus sitting still is still expending considerable energy on maintaining color and texture displays, processing sensory input from thousands of chemoreceptive suckers, and running the semi-autonomous neural networks in its arms. Feeding frequently is therefore a biological necessity rather than simply a behavioral preference, and octopuses are consistently described as voracious predators that spend substantial portions of their active period hunting. The three hearts, the hemocyanin-loaded blood, and the active lifestyle all form a physiological package that is internally consistent but operates at an intensity unusual for an invertebrate. The lifespan of most octopus species is surprisingly short, given the complexity of the animal. Most live between 1 and 2 years, with some larger species reaching 3 to 5 years. This brevity is partly a consequence of the reproductive strategy. Most octopus species are semelparous, meaning they reproduce once and then die, with the female investing her remaining metabolic resources entirely in brooding her eggs until they hatch and then declining rapidly. The three hearts, the blue blood, the distributed brain, all of this extraordinary biological machinery is built into a body that has, by evolutionary design, a planned obsolescence built into its life history. The octopus burns brilliantly and briefly, powered by blue blood through 3 tireless hearts, and the intensity of its living seems almost proportionate to its brevity. The skin of an octopus is a display technology more sophisticated than anything human engineering has yet managed to replicate. And the speed and precision with which it operates continues to amaze researchers who have studied it for decades. In less than a second, an octopus can transform its skin from a flat, uniform brown to a complex multicolored pattern featuring dozens of distinct elements, including spots, stripes, bars, eye spots, and textured three-dimensional projections called papillae, each element independently controlled and capable of being modified in real time. The control system that produces these displays operates in parallel at multiple spatial scales simultaneously, from the individual chromatophore cells up to the large-scale pattern organization that coordinates thousands of cells into coherent shapes. The primary color-changing cells in octopus skin are chromatophores, each of which contains a sac of pigment granules surrounded by a ring of muscle fibers. When the muscles contract, the sac is stretched flat and the pigment disperses across a wide area, making the cell appear colored. When the muscles relax, the sac retracts into a small, nearly invisible dot, and the pigment concentrates in too small an area to contribute meaningfully to the skin's apparent color. Each chromatophore is directly innervated by a motor neuron from the brain, giving the nervous system direct and independent control over each individual cell. With hundreds of thousands of chromatophores in the skin, the octopus has, in effect, a direct neural connection to each pixel of its body surface, a display system wired at the finest possible resolution directly to the brain. Beneath the chromatophore layer lie additional optical structures that complement the pigment-based color changes. Iridophores, similar to those found in chameleon skin, contain stacks of thin protein plates that interfere with light to produce structural colors, including blues, greens, and silvers that no pigment alone could generate. In some species, photophores, cells capable of producing their own bioluminescence, add a further layer of light production to the skin's optical repertoire. Below all of these lie leucophores, cells packed with white reflecting protein granules that scatter light diffusely and provide the bright white background against which the chromatophore colors are displayed most vividly. The layering of these different cell types produces a skin that can modulate not just color, but brightness, contrast, reflectivity, and in some contexts, polarization of reflected light, each dimension contributing to the complexity of the final display. The texture of the skin is independently controllable from its color, and the combination of the two produces displays of remarkable naturalistic complexity. The papillae, the three-dimensional skin projections that can be erected and retracted rapidly, are controlled by a separate muscular system from the chromatophores. When a resting octopus encounters a rough, irregular substrate like a rocky reef or a barnacle-covered surface, it can simultaneously adjust its color to match the coloration of the surface and erect its papillae to mimic the three-dimensional texture, producing a camouflage display that fools not just the color vision of a predator, but its perception of surface texture and depth. Researchers studying octopus camouflage have identified three broad pattern types: uniform, mottled, and disruptive. These are deployed in different contexts, depending on the background complexity, and the switching between these types is itself a rapid, contextually appropriate response. One of the most surprising discoveries in octopus visual biology is that octopuses are colorblind in the conventional sense of having only a single type of photoreceptor in their eyes, which means they cannot distinguish wavelengths of light the way animals with multiple receptor types can. And yet they produce elaborate, chromatically varied camouflage that appears to match the colors of their surroundings with impressive accuracy. How does a colorblind animal produce color-matched camouflage? Several hypotheses have been proposed to explain this apparent paradox. One involves the use of the pupil's unusual W-shaped or U-shaped form, which in a process called chromatic aberration allows different wavelengths of light to focus at slightly different depths in the eye, potentially allowing the single receptor type to sample different wavelengths at different pupil apertures as the focus is adjusted. A second, more provocative hypothesis proposes that octopus skin may itself be light-sensitive. Researchers discovered that opsins, the light-sensitive proteins that form the basis of photoreceptor function in eyes, are expressed in the skin of octopuses, cuttlefish, and squid, in the very chromatophore and iridophore cells that generate color displays. If these skin opsins are functionally active, the octopus may be sampling the wavelength composition of ambient light directly through its skin, and using this information to tune its camouflage display, bypassing the eye entirely for color-matching purposes. The idea that an octopus might see with its skin remains one of the most tantalizing and not yet fully resolved questions in cephalopod biology, and the implications of it for what it means to see, to perceive, and to be an octopus moving through a colored world it cannot see in the conventional sense. When an octopus extends an arm into the darkness beneath a rock, it does not know what it will find. The arm itself, with its 450,000 or so neurons running the full length, and its suckers each housing chemoreceptors that can simultaneously touch and taste a surface, will discover what is there and respond to it locally before any report reaches the central brain. This is not a limitation of the system. It is a feature, one that allows the octopus to conduct a sensitive, nuanced tactile and chemical search of a complex three-dimensional space without requiring the central brain to manage the process in real time. The arm searches, the brain does something else, and the arm will not bother the brain unless it finds something worth reporting. The suckers of an octopus arm are among the most complex sensory motor structures found in any invertebrate. Each sucker is a muscular cup capable of generating suction pressures strong enough to hold prey securely, or to anchor the arm firmly to a surface against considerable force. The rim of each sucker is densely packed with mechanoreceptors sensitive to touch and deformation, and with chemoreceptors, proteins that bind specific molecules from the environment and generate neural signals encoding chemical identity. This combination means that each sucker is simultaneously a grip, a touch sensor, and a taste organ. As an arm explores a surface, the suckers are continuously sampling both the mechanical texture and the chemical composition of what they touch, building a detailed profile of the surface that no purely visual inspection could produce. The spatial resolution of octopus sucker chemoreception has been tested experimentally, and the results suggest an impressive discriminatory capacity. Octopuses can distinguish between surfaces treated with different chemical compounds by touch alone, in the dark, without visual input. They can identify prey species, distinguish between edible and inedible items, and in some experiments appear to recognize previously encountered objects through their chemical signature. This chemotactile sense gives the octopus access to a dimension of information about its environment that vision cannot provide, and it operates continuously and passively, requiring no active decision to engage, simply the ongoing contact of suckers with whatever the arm happens to be exploring. The motor patterns generated by the arm's local neural network have been studied in detail, and they reveal a level of sophistication that goes well beyond simple reflex. When an octopus arm reaches toward a target, it does not extend in a straight line as a rigid rod would. Instead, it propagates a bend wave from the base toward the tip, a traveling deformation that allows the arm to navigate around obstacles, adjust its trajectory mid-reach in response to sensory feedback from the advancing suckers, and arrive at the target from a direction determined by local geometry rather than central planning. This traveling bend wave is generated by the arm's intrinsic neural network without requiring moment-to-moment instruction from the brain, and its parameters, speed, curvature, direction of travel, are modulated by sensory feedback from the suckers in a continuous closed-loop control process. There is a broader philosophical point embedded in the biology of the octopus arm. The standard model of vertebrate nervous systems assumes a relatively clear hierarchy. The brain is where intelligence lives, and the body is where it is expressed. The octopus challenges this assumption in the most direct possible way by distributing a substantial portion of its neural processing capacity into the body itself. If intelligence is understood as the capacity to process information and generate adaptive responses, then a significant fraction of octopus intelligence lives not in the brain, but in the arms. This raises genuinely interesting questions about what the octopus experiences, whether there is a unified experiencer receiving the integrated outputs of all this distributed processing, or whether the experience of being an octopus is something more fragmented and plural than our introspective categories can easily accommodate. The arm also plays a role in octopus rest behavior that adds another layer of interest to its semi-autonomous status. Researchers who have observed resting octopuses closely have noted that while the central body appears to be in a reduced activity state, individual arms sometimes continue to make small exploratory movements. A gentle probing of the substrate, a slow retraction or extension, a brief sucker grip test. These movements do not appear to be coordinated with any behavioral goal of the resting animal. They seem to be generated by the local arm networks operating quietly in the background, exploring the immediate environment at low intensity, while the central brain is otherwise occupied with whatever the octopus does during rest. The arm, it seems, does not fully sleep, even when the rest of the animal is resting. Most octopus species live for only 1 to 2 years, and the brevity of that life is not accidental, but deeply embedded in their reproductive biology. The majority are semiparous, a life history strategy in which the animal invests everything in a single reproductive event and then dies, channeling all remaining metabolic resources into reproduction rather than continued survival. This strategy is rare among vertebrates, where multiple breeding seasons spread across a longer lifespan are the norm, but it appears repeatedly among invertebrates in forms that make evolutionary sense under specific ecological conditions. For the octopus, which lives in a world of intense predation pressure and unpredictable food availability, the calculus of similarity appears to work. Breed once, breed maximally, and let the offspring carry the genetic legacy forward, rather than gambling on a second season. The reproductive biology of octopuses is extraordinary in several respects. Males possess a modified arm called the hectocotylus, one arm that has been adapted for the transfer of sperm packets called spermatophores to the female. In the act of mating, the male inserts the hectocotylus into the female's mantle cavity and transfers spermatophores that the female can store in specialized pouches for extended periods before fertilization. Some male octopuses, particularly in the genus Argonauta and certain deep-sea species, practice a form of mating in which the entire modified arm detaches from the male's body inside the female's mantle cavity, remaining functional and continuing to release sperm over time. Early naturalists who discovered these detached arms inside female octopuses assumed they had found a new species of parasitic worm, an understandable, if striking, misidentification. Female octopus brooding behavior is one of the most behaviorally intense examples of parental investment found among invertebrates. After laying her eggs, which she attaches in clusters to the walls of her den, the female enters a brooding phase in which she remains with the eggs continuously, aerating them by directing water currents across them with her arms, removing debris and potential pathogens with her suckers, and defending them against any intruder. During this brooding period, which can last from weeks to months depending on the species and water temperature, the female stops eating. She is drawing down her own body reserves, metabolizing muscle protein and energy stores accumulated before laying, to fuel the continuous muscular activity of brooding while receiving no caloric input. By the time the eggs hatch, the female's body is severely depleted, and she typically dies shortly after the last larvae emerge. The duration of the brooding fast and the depth of the female's commitment to it varies with water temperature in ways that reveal something about the underlying physiology. In cold water, development proceeds more slowly, and the brooding period can extend to several months. The longest recorded octopus brooding period is held by Granulodon boreopacifica, a deep-sea species living in cold water near the Monterey Canyon off California, where a single female was observed on multiple remotely operated vehicle dives over a period of 53 months, more than 4 years, continuously brooding the same clutch of eggs without eating. This observation, published in 2014, is the longest known period of dedicated egg care recorded for any animal. The female was visibly deteriorating across the observation period, but remained in position until the eggs hatched and then disappeared, presumed dead. For male octopuses, the post-mating period involves a different kind of decline. After mating, many male octopuses enter a state called senescence, characterized by loss of motor coordination, reduced responsiveness to stimuli, disrupted skin patterning, and eventually death. This senescence appears to be hormonally programmed, driven by the secretions of optic gland tissue that in female octopuses are associated with the commitment to brooding. In males, an equivalent hormonal shift appears to occur after mating, directing the animal toward a deteriorating terminal state rather than continued life and possibly further mating opportunities. Researchers who experimentally removed the optic glands from female octopuses found that the females stopped brooding and started eating again, even abandoning their eggs, and in some cases lived for considerably longer than unmanipulated females, suggesting that the senescent death of both sexes is at least partly under active hormonal control, rather than simply the inevitable consequence of metabolic exhaustion. There is something about the octopus's brief, intense life that carries a quality of completeness despite its brevity. The complexity of the brain, the sophistication of the skin, the intelligence of the arms, the extraordinary brooding dedication of the female. All of these are packed into a life measured in months, lived at high intensity, and concluded according to a biological program written into the animal long before it was born. From outside this life history, it can look almost tragic. From within the evolutionary logic that shaped it, the short, complete, maximally invested life of an octopus is a perfect solution to the specific survival and reproductive challenges of being an octopus. The briefness is not a flaw. The octopus has become one of the canonical examples of invertebrate intelligence in popular science, and the most frequently cited demonstration of this intelligence is the jar opening test. An octopus presented with a glass jar containing a food item inside and secured with a screw lid will, within a trial or few, solve the problem by grasping the lid with its suckers and rotating it counterclockwise until the jar opens. This behavior, requiring the coordination of multiple arms, the maintenance of goal-directed effort across the multi-step problem, and the spatial reasoning needed to understand the relationship between the lid's rotation and the jar's opening, is genuinely remarkable for an animal whose nervous system differs so fundamentally from those of the mammals and birds in which complex problem-solving was previously thought to reside. The jar test is impressive, but it is only one of a growing catalog of cognitively demanding tasks that octopuses have been shown to solve in laboratory settings. Octopuses can navigate mazes, including mazes with multiple choice points, and retain the learned solution across time gaps of days to weeks. They can learn to distinguish between objects differing in shape, color, or texture, and respond differently to each. They can learn through observation, watching another octopus perform a task and subsequently performing it more efficiently than naive individuals who did not observe the demonstration. This observational learning, sometimes called social learning, was for a long time thought to require the kind of social cognitive apparatus found in primates and other highly social species. Its demonstration in octopuses, which are largely solitary and have no social group to learn from in natural settings, was unexpected and has prompted reconsideration of which cognitive prerequisites are actually necessary for this kind of learning. Play behavior, which most behavioral researchers treat as a marker of a mind sufficiently complex to engage in non-functional exploratory activity, has been documented in octopuses in captivity. In a study conducted at the Seattle Aquarium, octopuses repeatedly pushed small pill bottles into the water current from the tank's filtration system, allowing them to be carried across the tank, retrieving them, and releasing them again into the current. This behavior served no apparent functional purpose, was not associated with feeding or predator avoidance, and was repeated consistently by the same individuals across multiple sessions. The researchers who documented it described it cautiously as play, acknowledging the interpretive difficulties, while noting that the behavior met the standard criteria of being voluntary, intrinsically motivated, and not immediately goal-directed. Individual personality differences in octopuses have been documented across multiple species and multiple research groups, and their existence has become one of the more scientifically interesting aspects of octopus behavioral biology. Individual octopuses show consistent differences in boldness, the willingness to explore novel environments or approach unfamiliar objects, in reactivity to disturbance, and in what researchers describe as general activity levels. These individual differences are stable across time and contexts, meeting the standard behavioral criteria for personality. Some individuals are consistently exploratory and approach new objects quickly. Others are consistently shy and retreat to their dens when disturbed. These differences are not explained by body size, age, or recent feeding history, and they appear to reflect genuine variation in the underlying neural or hormonal architecture of different individuals. The tool use of octopuses, while not as elaborate as that of some vertebrates, provides another window into the flexibility of their cognition. The most widely discussed example involves the veined octopus, Amphiooctopus marginatus, which has been observed collecting coconut shell halves discarded by humans, carrying them across the seafloor, and assembling them into a makeshift shelter at a new location. This carrying behavior is significant because the shells are cumbersome, provide no immediate benefit during transport, and are only useful when assembled at the destination. An animal that collects and transports an object for future use is engaging in a form of deferred planning that requires holding a future goal in mind during a current behavior that contributes no immediate reward. The documentation of this behavior in wild octopuses generated considerable scientific discussion about the cognitive requirements for what had previously been considered uniquely vertebrate-associated planning behavior. The intelligence of the octopus raises a question that comparative neuroscience is still working through. What is the minimum neural architecture necessary for complex, flexible, goal-directed cognition? The vertebrate brain, with its laminated cortex and extensive interconnection between specialized regions, has been treated as the gold standard for intelligent behavior. The octopus achieves many of the same behavioral outcomes with a radically different neural organization. No cortex, a distributed rather than centralized architecture, a brain that sits in a ring around the esophagus. If the same cognitive outcomes can be produced by such different neural structures, then the specific architectural features of the vertebrate brain cannot be the necessary substrate of cognition. The octopus is, in this sense, an existence proof that the vertebrate way of being intelligent is not the only way. An octopus hunting on a rocky reef at night is one of the ocean's more efficient and elegant predatory performances, conducted almost entirely in silence through a combination of visual tracking, chemotactile probing, and speed of execution that leaves most prey with almost no opportunity for successful escape. The octopus typically moves by crawling across the reef's surface, using its arms to pull itself forward, while its eyes scan the surrounding water and substrate for movement. When it detects prey, either visually or through the chemical trail left by a crab or fish moving across the substrate, it shifts from the slow exploratory crawl into a more directed approach, compressing its body, coordinating its arms into a more streamlined configuration, and closing the distance with an acceleration that its soft-bodied appearance does not immediately suggest. The strike itself can take several forms depending on the prey type and the context. The most dramatic is the pounce, in which the octopus launches itself onto a crab or fish, spreading its web between the bases of its arms to create a tent-like enveloping structure, then bringing all 8 arms underneath to grip the prey simultaneously with hundreds of suckers. This webbed pounce is extremely difficult for prey to escape, because the sucker contact is immediate and from all directions, and the enclosed space prevents the prey from generating the escape velocity needed to break free. Once secured, the octopus brings the prey to its mouth, which is located at the center of the arm crown, where a hard, parrot-like beak, hidden within the soft tissue, delivers a powerful bite that can penetrate hard crab shells. The beak is just the beginning of the octopus's prey processing capability. Behind the beak is a tongue-like structure called the radula, covered with rows of small teeth that rasp and pull tissue inward. And in the salivary glands adjacent to the mouth are produced compounds that are directly injected into the prey through the bite. These include paralytic toxins, primarily tetrodotoxin or related compounds in some species, and digestive enzymes that begin breaking down the prey's tissues from inside before the octopus has even started eating externally. A crab bitten by an octopus and injected with paralytic saliva typically stops moving within seconds. In some species, the octopus may begin drilling through the shell of a clam or crab using the radula before injecting saliva, using the tooth-covered tongue as a precision tool to create an access point for the chemical cocktail. The blue-ringed octopuses, a group of small species found in the Indo-Pacific, represent the most extreme development of octopus venomous capacity. These animals, which are among the most recognizable octopuses due to the iridescent blue rings that flash across their skin when they are disturbed or aroused, produce tetrodotoxin in high concentrations in their salivary glands, a neurotoxin that blocks sodium channels in nerve membranes and rapidly causes paralysis and respiratory failure. The toxin is produced not by the octopus itself, but by symbiotic bacteria living in the salivary glands, which the octopus hosts and presumably maintains as a chemical weapon manufacturing facility. A blue-ringed octopus small enough to sit comfortably in a human hand can produce enough tetrodotoxin to kill several adult humans, and there is no antivenom. The blue rings are a warning that virtually every other reef animal has learned to recognize and avoid. The intelligence of the octopus hunt extends to its opportunistic use of environmental features. Octopuses are known to follow other predators, particularly moray eels, which flush prey from crevices that neither predator could access alone. The eel drives prey out of a crack, and the octopus, waiting near the opening, intercepts what emerges. This joint hunting, sometimes described as coordinated interspecies foraging, was documented in the Red Sea in observations of octopuses actively approaching moray eels and apparently gesturing toward productive-looking reef structures before the two animals move toward the target together. Whether the octopus understands the complementary capabilities of the eel as a hunting partner, or whether this represents a lower-level learned association between eel presence and flushed prey, is an open question. Either interpretation requires a significant behavioral sophistication from an animal that has no natural social group to learn collaborative strategies from. After a successful hunt, the octopus typically retreats to its den to eat, bringing the prey inside and dismantling it within the shelter of the rocky refuge. Over time, dens accumulate midden piles of shell fragments, crab claws, and fish bones just outside the entrance, a visible archaeological record of the octopus's diet. Researchers can assess the hunting success and prey preferences of individual octopuses by examining these middens, and the composition of different dens often shows individual specific preferences that persist over time. One octopus may preferentially take a particular crab species. Another may focus on bivalves. These dietary specializations, which go beyond what can be explained by local prey availability alone, suggest that individual octopuses develop preferred hunting strategies over their brief lifetimes, refining and repeating approaches that have worked for them rather than treating each hunt as a fresh, unconstrained problem. The octopuses that most people know are the shallow-water, reef-associated species that have been accessible to researchers working with scuba and aquarium tanks for decades. But the ocean has a third dimension that extends far below the reefs into perpetual darkness, extreme cold, and pressures that crush unprotected equipment and make sustained human observation essentially impossible. In these deep and dark environments lives a diversity of octopus species that we have glimpsed only briefly and partially, through the occasional remotely operated vehicle dive and the examination of specimens caught incidentally in trawl nets. What has been glimpsed is often extraordinary. Deep-sea octopuses have evolved in the absence of light, and many of their adaptations reflect the particular challenges and opportunities of that environment. Some deep-sea species have reduced or absent chromatophore systems, since there is no light for color displays to function in, replacing the elaborate skin-based communication of their shallow water relatives with other signaling modalities, or simply dispensing with visual communication altogether. Some have evolved enhanced mechanoreceptive systems for detecting water movement and pressure waves, which remain effective in darkness. Others have evolved bioluminescence, producing their own light either for communication, prey attraction, or counter-illumination to hide their silhouette from below. The diversity of solutions in the deep sea is, as in so many contexts, a testament to what evolution can produce, given sufficient time and a sufficiently different set of conditions. The vampire squid, Vampiroteuthis infernalis, occupies the boundary between octopuses and squids in the cephalopod family tree, and lives in the oxygen minimum zones of the deep ocean, between 600 and 900 meters depth, where oxygen concentrations are so low that most animals cannot survive. Its metabolism is remarkably slow, and its oxygen requirements correspondingly reduced, allowing it to exploit a zone of the water column that is largely predator-free, precisely because so few animals can tolerate the conditions. Rather than actively hunting, it feeds primarily on marine snow, the continuous rain of organic particles, dead cells, and fecal matter that falls from the productive surface waters, filtering this material with long, filamentous arms covered with fine sensory structures. It is not a predator in any meaningful sense. It is a deep ocean scavenger and filter feeder in the body plan of the cephalopod, a reminder that the octopus body plan is more versatile than any single species suggests. Among the largest of the deep-sea octopus-like cephalopods is the giant Pacific octopus, Enteroctopus doflani, which is not truly a deep-sea species, but is the largest octopus known, with confirmed arm spans reaching up to 4 meters and body weights exceeding 40 kilograms. This animal's nervous system scales up in complexity with its body size, and captive individuals have demonstrated problem-solving and memory capacities that researchers describe as impressive even against the backdrop of octopus intelligence generally. Their size also makes them unusual in terms of lifespan. Giant Pacific octopuses can live for 3 to 5 years, considerably longer than most other octopus species, giving them more time to accumulate experience and potentially to develop more elaborate individual behavioral histories. The Dumbo octopuses, genus Grimpoteuthis, are among the most visually distinctive of the deep-sea species, named for the large ear-like fins that project from the sides of their bodies, and which they use as the primary means of locomotion, flapping them gently to swim in a way that bears no resemblance to the crawling of shallow-water octopuses. These fins give them an appearance sometimes described as endearing, and footage of them swimming in the likes of deep-sea vehicles has produced some of the more widely shared ocean exploration video content of recent years. Biologically, they are poorly known. Their diet, their behavior in the dark outside of vehicle lights, their mating and reproductive patterns, their cognitive capabilities, all of these remain largely unknown. They represent the majority condition for deep-sea octopus species, present, presumably significant components of deep-sea ecosystems, and almost entirely unstudied. The darkness of the deep ocean is not simply an absence of light, but a different ecological world operating on different rules, with different currencies of energy and information, different competitive pressures, different relationships between body plan and behavioral strategy. The octopuses that live there have evolved in response to these rules over millions of years, and what they have become reflects those rules as precisely as shallow water octopuses reflect theirs. We have seen enough of the deep-sea octopus fauna to know that it is diverse, ecologically significant, and often extraordinary. We have not seen enough to know what the most remarkable of them can do. The deep ocean keeps its secrets in the dark, and the octopuses that live there keep them too, going about whatever it is they do in the cold and the pressure and the permanent night, unobserved, and in the most literal sense, unknowing that anyone is curious about them. The octopus exists at the center of one of the more genuinely interesting philosophical questions in contemporary animal cognition research. What is it like to be an octopus? The question parallels a famous philosophical essay by Thomas Nagel titled, "What is it like to be a bat?" which argued that the subjective experience of an animal with radically different sensory systems and neural organization than our own is fundamentally inaccessible to human understanding. Not merely unknown in its details, but different in its very nature from anything we can imagine from inside our own perceptual world. The octopus, with its distributed nervous system, its skin-based sensory and display capacity, its chemotactile arms operating semi-independently, and its single receptor color vision experiencing a visual world whose chromatic information it processes through means we don't fully understand, presents at least as great a gap from human experience as the bat that inspired Nagel's original question. The question of octopus consciousness is taken seriously by a growing number of researchers, and it received an important formal acknowledgement in the Cambridge Declaration on Consciousness, a document produced by a prominent group of neuroscientists in 2012, which stated that the neural substrates necessary for conscious experience are not limited to the cortical structures found in vertebrates, and that non-human animals, including octopuses, possess the neurological structures that generate conscious states. This was a significant step, given that previous default assumptions in the field had tended to link consciousness tightly to the vertebrate brain architecture, and specifically to cortical processing. The declaration does not resolve the question of what octopuses experience, but it shifts the burden of proof toward those who would deny that they experience anything at all. The sleeping octopus provides one of the more recent and unexpected pieces of evidence relevant to the question of octopus inner experience. In 2021, researchers studying Octopus insularis in Brazil observed and filmed octopuses undergoing rapid, dramatic changes in skin color and texture during what appeared to be sleep, cycling through complex patterns across a period of seconds, then settling, then cycling again. The rapidity and dynamism of these color changes during apparent sleep, combined with the accompanying rapid eye movements visible through the closed eyelids, raised the possibility that octopuses experience something functionally analogous to REM sleep in mammals and birds, the sleep stage most associated with dreaming. If octopuses do experience something during this active sleep phase, what they experience, given the richness and complexity of their waking skin displays and sensory life, is a question that generates a kind of quiet wonder when you sit with it long enough. The solitary life of most octopuses raises interesting questions about their social cognition. Animals that live in persistent social groups, like primates, corvids, and dolphins, have been argued to have developed their advanced cognitive abilities at least partly in response to the demands of tracking social relationships, remembering individual identities, and past interactions, and navigating the complex social dynamics of group life. The octopus, being largely solitary, has apparently evolved comparable cognitive sophistication without this social driver, suggesting that the problem-solving and learning demands of the physical environment alone are sufficient selective pressure to produce complex cognition. This has implications for thinking about the origins of intelligence more broadly, and for considering whether social complexity is a necessary condition for cognitive complexity, or merely one of several possible paths to the same outcome. Some octopuses have been observed behaving in ways that have no obvious functional explanation, and that some researchers have interpreted as indicating something closer to emotional states than simple stimulus-response reactions. An octopus disturbed from its den by a diver may perform what looks like an elaborate threat display, spreading its arms, flashing color patterns, and advancing toward the disturber before retreating. This behavior seems to exceed what pure predator avoidance would require, and its escalating theatrical quality has led some observers to describe it as something like anger. Whether any emotional label is scientifically appropriate for an octopus reaction is deeply contested, but the behavioral observations that motivate such descriptions are real, and they contribute to a growing body of evidence that the octopus's internal states are more complex than simple reflex would account for. What the octopus ultimately represents, in the broadest sense, is an independent answer to a question that evolution has posed multiple times. What can complexity of mind look like? And how many different architectures can support it? The vertebrate answer, refined over 500 million years, is well known to us because we are inside it. The octopus answer, refined over the same period from a completely different starting point, using a completely different neural organization, distributed differently through a body built on different principles, is still being read. And the more carefully we read it, the more it seems to tell us not just about octopuses, but about the nature of mind itself, and about how many different ways the universe has found, in its long and patient experimentation, to arrange matter into something that looks outward at the world, and in some sense we do not yet fully understand, wonders. The reef changes when daylight fades. The species that are visible and active during the day retreat to crevices and burrows, and a different community of animals emerges to exploit the darkness. The octopus is among the most significant of these nocturnal operators, moving across the reef with a freedom it does not have during the day when visual predators are active, conducting the majority of its hunting in the hours between sunset and sunrise, when it is most difficult to detect against the complex, shadowed background of the reef. On a quiet night reef, the octopus is often the most active large predator moving across the rubble and coral, and its influence on the reef's prey community, the crabs, mollusks, small fish, and other invertebrates it consumes, is disproportionate to its modest body size. The use of den sites by octopuses imposes a spatial structure on their activity that has ecological consequences for the reef community around each den. An octopus in residence at a particular rock crevice will repeatedly hunt the surrounding area, depleting preferred prey species within its regular foraging radius, and forcing the prey community around the den to restructure over time towards species or size classes less preferred or less accessible to the resident octopus. When the octopus dies or moves on, the prey community around the abandoned den gradually recovers. This local ecological cycling, driven by the hunting pressure of individual octopus residents, contributes to a patchy, heterogeneous prey distribution that characterizes many reef communities. The octopus is not simply a consumer in this system. It is, through its spatial and behavioral ecology, a sculptor of the community around it. The camouflage of the octopus on the reef at night is of a different quality from its daytime concealment. At night, with no visual predators actively hunting by sight, the functional demands of camouflage change, and many octopuses adopt relatively simple, dark colorations that minimize their visibility in the low light, rather than the elaborate background-matching displays they produce during the day. Some researchers have observed that resting octopuses on the reef at night maintain a degree of skin pattern activity, even when apparently unaware of any observer, cycling through simple pattern elements in a subdued, unhurried way that looks less like active camouflage management and more like the low-level expression of a system that is never entirely quiet. The relationship between octopuses and the moray eels that share their reef habitat is one of the more ecologically interesting interspecies relationships in the reef community. The two animals occupy overlapping but not identical niches. Both are predators of crabs and other invertebrates, but they access prey through different means. The eel through its elongate body and flexible jaw pursuing prey into crevices, the octopus through its flexible arms and suction exploring different geometries of shelter. The joint hunting behavior observed in some populations appears to be a learned, individually variable strategy rather than a fixed, species-typical behavior, with some octopus individuals consistently following eels and others not, and with the effectiveness of the strategy depending on the specific behavioral tendencies of both the octopus and the particular eel involved. The octopus's relationship with its own den is worth reflecting on briefly. The investment in selecting, maintaining, and defending a den site suggests that the octopus has something like a home in a psychological as well as a spatial sense. Individuals return to the same den repeatedly over days and weeks. They maintain it, removing debris and unwanted occupants with their arms. They defend it against rivals and potential predators with a consistency that suggests the den's value to them exceeds any momentary cost-benefit calculation about the specific encounter at hand. Some researchers have noted that octopuses appear to recognize their own den from a distance, approaching it directly through apparently learned spatial memory of its location, rather than by random searching. The den is home, in the sense that the octopus knows where it is and returns to it by choice, which is a quietly significant thing for an animal with a one-year lifespan and a nervous system built on entirely different principles from our own. As the night advances and the reef settles into its deepest quiet, the octopus continues its slow, considered progress across the substrate, reading the reef through its suckers, watching through eyes that can see in near darkness, thinking through a brain distributed between a central ring around its esophagus and the 8 arms trailing behind and before it in the current. It does not know it is extraordinary. It does not know that scientists have been trying to understand it for decades with incomplete success. It simply moves through its world with the fluid, purposeful intelligence of an animal that has been very good at being what it is for a very long time. And the reef holds it gently, the way it has always held the octopuses that have lived on reefs like this one since before there were humans on the land, above the water, to wonder about what moved in the dark below. And so the current carries us gently back towards stillness. The reef is quiet now in the deep hours of the night, and somewhere on the dark substrate below the surface, the octopus has returned to its den. Arms pulled in close, skin settling into its resting pattern, 3 hearts beating their blue-blooded rhythm in the cold. At rest in the way that something with arms that never fully stop is ever fully at rest. You have spent tonight with one of the most genuinely different minds our planet has produced. A mind that sees through skin as well as eyes, that thinks through arms as well as brain, that is brief and brilliant and wholly itself in ways that our vocabulary for intelligence barely reaches. There is something strangely comforting in that difference, in the knowledge that the world contains minds shaped on principles so unlike our own, thriving in the dark water in ways we are still learning to see. Let the room hold you now, the way the water holds the octopus, softly, fully, without effort. The thinking can rest, the wondering can pause, the arms can be still for a while in the quiet.